Hi, everyone. Good afternoon to those of you in person as well as those over Zoom. And today is our last mental health and addiction rounds for this term. We'll come back in September with an exciting lineup as well. But today it's my pleasure to present Dr. Shyh-Dar, who is the Angiotech Professor in Drug Delivery at the UBC Faculty of Pharmaceutical Sciences. His research focuses on developing innovative drug delivery systems to enable novel therapies, including biological drugs and nanomedicines. And today he will present KK-103, a novel prodrug of leu-enkephalin for antinociception and mood improving. So please join me in welcoming Dr. Lee.
Thank you so much. And thank you, Tony, for this wonderful opportunity to share my work with you. So as Lily just said, my lab is actually working on drug delivery systems. Yeah. And so this is sort of in the junction of prodrug and drug delivery, because we are modifying a parent drug produced in our body to make it available to our body. Let's see if I can advance the slide. Okay, let's try. Okay, good. So, topic is about chronic pain and depression. So you all know very well about chronic pain, but basically, from my understanding, it's defined as pain persisting for more than 3 months. So it's actually quite prevalent in Canada and globally. So about 20% of Canadians are experiencing chronic pain right now. And it comes with a lot of comorbidity. So depression is one of them. As you can imagine, people who are under pain probably are not very happy. So 60% of them also suffer from depression. So we are thinking whether there's a single drug that can address both problems. And as I said, this is chronic pain is a really significant problem, has a lot of societal impact right now. First, it affects the physical and the psychological state. It affects productivity, of course, and it also decreases quality of life of many patients. So this is a very significant problem. And we have lots of drugs available to treat either these two, but none of them are perfect right now. So this is a very high-level overview of our current pharmacological treatments available for chronic pain. And so they are delivered based on the severity of the pain. For example, usually we start with the mild cases and the moderate effective drugs like NSAID or acetaminophen. For more moderate cases, then we probably need to advance to topical anesthesics like lidocaine or capsaicin for more localized pain. And if the pain is associated with neurological involvements, and then antidepressants like tricyclines and also serotonin reuptake inhibitors, or even anticonvulsants will be used, such as gabapentins. And if the pain is really severe, then we will consider opioids, starting from weak opioids all the way to stronger opioids like morphine, fentanyl. And obviously, cannabinoids are also one of the hot, new drugs that are being investigated for more severe chronic pain. So as I mentioned, opioids are one of the most effective drugs for chronic pain, especially in severe cases. So their chemical structures are largely in this, this structure, and with some modifications to adjust their potency. And so as I mentioned, there are some famous examples: morphine, oxycodone, and codeine. And but they are very selective for mu opioid receptor. but we also have delta and kappa. And they activate mu opioid receptors that comes with a lot of effects in addition to analgesic effect. So those include tolerance, dependence. So that will make patients take more and more drugs to maintain that same level of efficacy. And over time, that could lead to overdose. And respiratory depression is the lethal side effect, as you are very aware of. And also sedation and constipation, they are also common side effects coming from opioids. So opioids are addictive and cause severe adverse effects. And so we have quite a bit of opioid overdose death, especially during and after pandemic. This problem is devastating our health system. Okay, so our body actually also produces opioids, endogenous opioids. They are coming as this peptide form. So we have several peptides that are opioid neurotransmitters, including beta endorphin. We have enkephalin, and we also have dynorphin. So these peptides, they are actually activating different opioid receptors, more selective towards one over others. For example, endorphin activates mu, and then it comes with those good effects and not so good effects, such as respiratory depression, constipation, as we just mentioned, in addition to euphoria and analgesia. But for leu-enkephalin is very selective for delta, and it mainly induces those so-called beneficial effects such as euphoria and analgesia. And dynorphin activating kappa, as you can see, is this dysphoria, which is not so desirable. So in our lab, we focused on leu-enkephalin, develop prodrugs or delivery technologies to improve the delivery of leu-enkephalin into the brain or the affected sites to have desirable effects. So as you probably can appreciate, the native leu-enkephalin structure here is a 5-amino acid. So for amino acid drugs, they comes with a lot of problems. First, they are not stable. Lots of enzymes could digest them. So if you introduce them peripherally into our body, it's not going to be effective to induce any measurable pharmacological effects. And it also is not membrane permeable. So in this case, leu-enkephalin probably is not the worst case, but again, it carries two charged. First is at the N-terminal is amino group there. And at the end, it has a carboxylate group. So it carries two charges per molecule. So those our cell membranes don't like those charged molecules. So they have a very poor membrane permeability. So that limits their systemic absorption and permeation into our brain to have any effect. So in a lot of literature, when people use leu-enkephalin or develop prodrugs or derivatives of leu-enkephalin, they usually introduce these drugs through direct brain injection instead of those commonly used drug delivery routes such as subcutaneous delivery or oral absorption. So we are not the first group interested in leu-enkephalin. So a lot of labs actually produced leu-enkephalin derivatives or methionine enkephalin derivatives. So their approach is usually introduce D-amino acids, right? So that will stabilize or increase the stability of the parent or native peptide. They also modify some of the chemical structures to make it either more stable or more membrane permeable. So in this case, in the first case, they introduced this amide group at the end of the carboxylate tail to improve the stability. They also incorporate a D-amino acid here and replacing the glycine, second amino acid in the leu-enkephalin using D-alanine. So a lot of other modifications you can see here. They are all doing using similar approaches to improve stability, membrane permeability. And even in this case, it cyclized the peptide to make it stable. But all these drugs, they still require direct brain introduction. So they are not bioavailable after peripheral delivery. And sometimes they also introduce significant side effects. In many cases, they observed convulsion in mice, in rodents. So our approach is a little bit different from the previous approach. So we want to preserve the native structure of leu-enkephalin and only introduce a very minor modification to the N-terminus. And this N-terminus modification is only coming from very minor, small N-terminal chemical groups, right? So these are a few of the chemical structures we introduced to the N-terminus. So they are all very small. So the purpose is that we want to preserve the native leu-enkephalin structure as much as we can so that we are not introducing all other side effects, possible side effects. But in the meantime, we are hoping this N-terminal modification can improve stability of the peptide as well as membrane permeability of the peptide, because it blocks the amino group, the charge, at least one of the charges that leu-enkephalin carries. And then we follow by a few assays that we could screen these compounds, but we quickly went into in vivo efficacy study. And then very luckily, we got one hit. We call it KK-103. So the chemical structure of KK-103 is shown in here. It's basically a pivalic acid conjugation to the N-terminus. And then this is the structure here. So I think in the following few slides, I'm going to introduce the efficacy, safety, pharmacokinetics, and also mechanism of KK-103 that we have studied over the past few years. So the first study we did was this hot place test. So you know this very well, so I don't need to introduce this method to you. And we monitor the antinociception effect over time after drug administration. So in here, we compared 3 drugs. The first one, KK-103 blue, and we have morphine in red, and we have native leu-enkephalin in black. So as you see that leu-enkephalin delivered by subcutaneous injection is not effective at all. Morphine is very effective, and the onset is really fast. 30 minutes, we hit the peak, but it declined very fast as well. So within 2 hours, it's all gone. And KK-103, instead, the effect is sort of delayed. We started to see significant analgesic effect at 1.5 hours after subcutaneous injection, and it peaks at 2 to 3 hours and slowly decline to 5 still seeing some significant effect at 5 hours. So it's a slow onset drug, but with some prolonged activity. So this is sort of what we expected because this is a prodrug. It takes time to be converted back to the parent drug. So that's one model, and we also did another model. This is formalin pen injection formalin injection model. Again, you're probably familiar with this. Basically, we give the drug subcutaneously, and then we inject formalin into their foot pad, and just monitor the licking and biting behavior of those mice. So we are looking at phase 1 and phase 2 responses. So my understanding is that phase 1 is usually coming from acute response. And then that usually, if the drug has some central efficacy, coming from the brain, then we should be able to see some efficacy. And the phase 2 is more information-induced behavior. And then if so the efficacy could be coming from the central as well as peripheral. If we focus on phase 1, you see that morphine and KK-103, they were equally potent in suppressing phase 1 response. And phase 2, morphine is super effective. It abolish those phase 2 responses. But KK-103 is also doing well. You see that the phase 2 responses are reduced to half compared to the vehicle control. And so I think in the first slide, we already showed this time course pharmacology. And this is sort of a repeat experiment with REM hot plate study. This is just a different type of hot plate. And so we got very comparable results. So again, we started to see significant effects after 1 hour. It peaks at 2 hours and then slowly decline, but it's still significant at 4 hours. But after 8 hours, it returns to baseline. So again, this is a delayed but also prolonged pharmacological effect from KK-103. And dose-dependent pharmacology is also very important. So we did both REM hot plate study. So in here, we compare a range of doses from 1 to 50 milligram per kilogram of KK-103. And so the data suggests that at 5, it started to become significant. And the effect seems to peak at 10. And then at 20 is still effective, but at 50, the higher dose, surprisingly, the effect is gone. So we will get back to this after we explain the mechanism later on with some data. So this is from the REM hot plate study. So another model, formalin food model, also gave us similar results. So we started seeing effects at 5, peaks at 10 to 20, but at 50 milligram per kilogram, the effect is gone. So very comparable results with both models. And how about sex differences, right? So in here, we look at the antinociceptive effect in both female and male mice. And so they both show significant effects after KK-103 subcutaneous injection. And there's no significant difference in terms of efficacy between female and male. But there tend to be a trend showing that female are more sensitive to this heat stimulus compared to male. But again, it's not really significant. Okay, so as I mentioned, leu-enkephalin is native peptide. It activates delta receptor primarily. So it also comes with euphoria effect. So we were curious about whether KK-103 can produce antidepressant-like behavior in animals. So in this case, mice. So this first experiment is called force swim test. And so my student recorded the immobility time as an indicator of so-called depressed state. So we have a negative control vehicle, and we also have a positive control despyramine. This is a very potent tricyclic drug, antidepressant drug. As you can see, it suppresses the immobility time very significant. And KK-103 is also doing a good job and is also significant compared to vehicle control. So another commonly used test to measure the antidepressant-like effect is called tail suspension test. Again, it's also to record the immobility time of mice. So we got very comparable results showing KK-103 also has some effects in suppressing this so-called depressed state of mice. So those look quite promising. So as we mentioned earlier that while the reason we want to harness our endogenous system to treat chronic pain as well as depression is that we believe it will be a safer approach. So the safety is the number one parameters we look at. So as we mentioned earlier, the respiratory depression or suppression is the number one side effects of opioids. So definitely this is a very significant or dose limiting toxicity we want to monitor. And so we compare vehicle control, we have morphine and we have KK-103. So morphine induces their effect as early as 30 minutes. So in here, you see that this respiratory suppression is very significant 30 minutes after subcutaneous injection of morphine. And KK-103, we appreciate that there's some delayed effect from KK-103. So we monitor respiratory or breathing rate over time at 30 all the way to 4 hours after subcutaneous injection of KK-103. And we didn't see any significant difference compared to the vehicle control. So another thing we want to look at is so-called tolerance. So meaning that opioid efficacy will go down over time. And indeed, this is the case for morphine. So morphine were given subcutaneously to mice daily for 7 days, and then we monitor the hot plate efficacy over time. And you see that on day 7, the efficacy, the overall antinociception effect of morphine has decreased significantly. So for KK-103, this effect is very mild and is not significant. And so this is dependence, physical dependence, they say. So we subcutaneously inject morphine or KK-103 or vehicles twice daily for 7 consecutive days. And on day 8, we still give them the morning dose, but the afternoon dose, we skip that, and instead we IP gave a naloxone, which is a opioid antagonist. So that will block all the opioid receptors to induce opioid withdrawal. So those mice, if they become physical dependent on opioid or whatever we give to them, they will show unease behaviors. So in this case, jumping. So we counted number of jumps of those animals, the first 10 minutes after naloxone injection. So only morphine is showing very significant dependence, not KK-103 or vehicle. So constipation is another commonly seen side effect, and although it's not lethal, is really causing a lot of discomfort to patients. So in this case, we did this so-called charcoal meal assay. So we during the treatment with either vehicle, morphine, or KK-103, we give animals charcoal meal. So those charcoal is black, and then you can follow and quantify their GI transit distance. And then that will show the mobility of the GI tract. So that is an assay for us to quantify constipation. So this is vehicle control, and so the gastrointestinal GI transit rate is close to 80%. And morphine is very significant in terms of suppressing this GI transit rate down to 20%. KK-103 is doing much better compared to morphine. It hits 50%, but it's still significant compared to vehicle. So there's some side effects coming from constipation, but not as severe as morphine. My student also looked at the so-called sedation effect. And the way he did it was put mice in an open field and then basically measuring their reeling behavior, right? So in this case, morphine mice show decreased reeling behavior, but KK-103 didn't show that. Okay, so we have data suggesting that KK-103 is efficacious and safer compared to morphine. But now we want to go back to understand the mechanism. First, as we hypothesize, conjugating a pivalic acid to the N-terminus of leu-enkephalin should be able to sort of decrease the enzymatic digestion by endogenous enzymes in our body. So that should be able to increase the stability in different biological fluids. So that's one hypothesis. And let's look at the data. So this is the mouse plasma, so in vitro. So we dissolve KK-103 or leu-enkephalin in black in this case. In mouse plasma, and over time we take out the sample and then did HPLC analysis. So as you can see, leu-enkephalin is very susceptible to enzymatic digestion in the mouse plasma. So we think the half-life for leu-enkephalin is only 25 minutes. And but for KK-103, you see that it remains stable, more than 90% at five hours after incubation. So the enzymatic stability of KK-103 is significantly improved compared to the native peptide. So we were also wondering that if the compound survived plasma, how about the brain conditions, right, or the central nervous system conditions? So we incubated these two compounds again in CSF. And so in CSF, because the enzymatic concentration is much, much lower, about 1/10 lower compared to plasma. So even leu-enkephalin shows quite reasonable stability, right? So 5 hours, we still have 70%. But again, this also shows that KK-103 is more stable compared to leu-enkephalin in a different biological fluid, CSF in this case. So I think this data also explains why the earlier study had to do direct brain injection to see leu-enkephalin efficacy and other derivatives developed early on. But in our case, it seems like KK-103 is able to survive plasma and CSF. So that also explains why we can do subcutaneous injection and still see some pharmacological effects. Okay, so we also study pharmacokinetics of KK-103, and this is comparing KK-103 and leu-enkephalin. And yeah, to my surprise, we did see some a very minor absorption of leu-enkephalin from subcutaneous injection. So it does survive, our body at some level. But as you see that the absorption is really slow, possibly because, again, leu-enkephalin's membrane permeability is low, so it doesn't pass through cell membrane or other membranes as effective as KK-103 to be absorbed effectively as KK-103. So if we look at KK-103, the diamond shape data here, you see that there's a very rapid absorption. We started detecting plasma concentration at 2.5 minutes after sub-Q injection and peaks at 5, and then but rapidly decline. But even at 1 hour after subcutaneous injection, we could still detect some level of KK-103. And definitely there is some dose dependency here. So as you can see, we increased the dose from 5, 10, 20, and then 50. And then you see that the peak, the plasma curve just getting bigger and bigger. So that's dose dependency. But we also noticed that the dose linearity only happens between 5 and 20. So not beyond that. So at 50 milligram per kilogram, the overall absorption or the overall drug exposure is not is not as high as projected. Okay, so this is because the KK-103 has a very low solubility. So in this case, when we had to inject 50 milligram per kilogram, we had to inject a large volume of medium under the skin, and that could affect the slow absorption. And even even maybe after 1 hour, there could be some residual dose not fully absorbed. So we were thinking maybe that's the reason that we didn't see this good linearity at 50 milligram per kilogram. Okay, so one thing I want to also let you know is that we compare different delivery routes for KK-103. This is IV and sub-Q. So the black line is IV. So again, it reaches the highest concentration at time 0 and then rapidly decline. And by KK-103, you see a absorption curve there, but still very quickly absorbed. But the decline is slower compared to IV. So this PK is called so-called flip-flop phenomena, but that's more for you to just to be aware of. But basically, this is to tell you that the absorption rate is slower than the elimination rate. So it has this kind of a phenomenon of delayed elimination for the subcutaneous injection compared to IV delivered compound. So from the comparison, the half-life for sub-Q is increased by 3-fold. The AUC area under curve to determine the overall drug exposure, I would say they are very comparable. This 40% difference is not considered significant for PK. studies. Yeah, so this is the reason we pick subcutaneous injection for most of our studies. Okay, so now we have this PK. We show that it can be systemically absorbed after subcutaneous injection. Another question is whether the compound can reach the brain. Because earlier we showed some efficacy data, right? This hot plate assay that usually those mice are normal mice, naive mice, and suggesting that the KK-103 must be acting in the brain to have that kind of efficacy. So are we delivering to the brain? So this is a tissue distribution study. So this is in collaboration with Urs Hafley in our faculty, and he helped us label KK-103 with a radioisotope. And then we deliver to mice, and we use imaging method. And we also collect individual tissues to analyze the radioisotope amount to measure the pharmacokinetics or the drug uptake in individual tissues. So this top panel shows tissue uptake in higher amounts, right? So from blood all the way to stomach, and from 1% to all the way to 9% injected dose per gram. And so one thing I want to show you is that the small intestine is showing a quite high uptake of KK-103. And I think that this explains why we are seeing this constipation side effects from KK-103, although it's reduced compared to morphine. And in the bottom, we are showing tissues that are displaying lower amount of KK-103 uptake from 0.1 to 0.8% injected dose per gram. And brain, we could actually detect some level of uptake. So my student also sectioned the brain, and then basically he's showing that indeed there's KK-103 going to the brain. Okay, so we were hypothesizing that KK-103 is a prodrug. So that's why we see this delayed and prolonged pharmacological effect. But how about direct evidence of data showing that it is indeed a prodrug? So this is a biological assay looking at delta opioid receptor activation, looking at the inhibition of CAMP accumulation in cells, expressing DOR. So we treat them with either leu-enkephalin or KK-103. And then you see this sigmoidal pharmacological curve so that you can get the effective concentration 50. And as you can appreciate that their activity is very different, right? So it's a 30,000-fold difference that basically you can consider KK-103 ineffective compared to leu-enkephalin. So this is indeed a prodrug because the prodrug itself is not working. It has to be converted back to leu-enkephalin to have the effect. So how about the release of the parent drug from KK-103? Earlier, we showed that even as CSF is not really releasing anything, right? KK-103 still remain intact at 5 hours after incubation. So we went back to the literature, and then some suggest that neuron cells express some enzymes at their cell membrane. So those could be effective in releasing KK-103. Releasing leu-enkephalin from KK-103. So in this case, we incubate leu-enkephalin with cells, and then we just measure the KK-103 and leu-enkephalin in the medium over time. So in the beginning, though, even without cells, there's some baseline release already. So we could detect some, even without salt, just in the medium. And then in the presence of salt, we can see that the release of insulin, actually release of enkephalin increases over time. And KK-103 decrease over time, suggesting that KK-103, leu-enkephalin is slowly released from KK-103 in the presence of cells. Okay, and definitely we really want to understand whether KK-103 is acting primarily on DOR. And so we actually did these experiments in vivo because for us, it's much easier to do in vivo study compared to in vitro engineering different cell lines. And so in this first study, we co-administered mice with KK-103 or vehicle with vehicle or some opioid receptor inhibitors, selective opioid receptor inhibitors, right? So in this case, we have a DOR inhibitor, selective again. It's not 100% specific, but it's very selective for DOR. It's nitrindo. We also have the so-called pan opioid receptor blocker, naloxone. So it will block all opioid receptors. So as you can see that after adding DOR inhibitor, the efficacy of KK-103 is reduced to the baseline compared to the vehicle control, and also the naloxone block group. So it looks like it's quite so DOR is the major receptor that KK-103 targets. And so this is another blocking assay. So we use selective MOR inhibitor, naloxone. And so again, it's significant. So, but it doesn't really completely block the KK-103 efficacy to the baseline. So suggesting that KK-103 has some activity for mu opioid receptors as well, but mu opioid receptor is not as important as delta. We got similar results as kappa receptor blocker, so very similar as morphine. Mu opioid receptor. So again, so the message is that antagonism with naloxone and nitrindo, which is a selective DOR blocker, almost completely blocks KK-103's effect. But when MOR or KOR are blocked, KK-103 show a partial effect. So this suggests that KK-103 is primarily acting on DOR. Okay, so I think this is just additional results that we got earlier. I think it's interesting to show that in this hot plate study, indeed, the KK-103 effect is coming from the central. So we have the naloxone, going everywhere in the body of mice, or naloxone methiodide, which is a charged analog of naloxone. It's not brain permeable. So it doesn't go to the brain to block opioid receptor. So in that case, it doesn't block KK-103's effect. So only the blocker that going to the central is able to block KK-103 effect in the hot plate study. But I think we now need to do a similar study in this formalin model, just to check in a different model, inflammatory pain model, are we seeing effects from peripheral as well? Okay, so quickly go through the conclusion. And KK-103 shows antinociceptive effect in nociceptive and inflammatory pain models, prolonged activity over morphine and antidepressant-like effects in mice. And compared to morphine, KK-103 induces fewer and weaker side effects. And KK-103 displays increased stability and improved absorption compared to leu-enkephalin. And KK-103 shows conversion to highly potent leu-enkephalin in the presence of neuron cells. And the antinociceptive effect of KK-103 is largely mediated by the DOR. So future plans, this is still a ongoing project in the lab. And so we are funded by MyTex as well as Health Research BC to basically examine the efficacy of KK-103 for treating opioid withdrawal symptoms. And this is in collaboration with Tony and Puya. And we also recently got funding from Arthritis Society Canada. And that project is to develop a topical formulation for KK-103 to treat OA pain, because we think that this is a so-called high-risk, high-reward project idea that maybe KK-103 is showing really good effect in inflammatory pain, and it could have some peripheral control of the pain. And so this is in collaboration with Brian Cairns. And recently, my new postdoc Brody just performed the first preliminary test just to examine whether KK-103 is skin permeable. So this device is called Francel. So we have a top channel, and we also have bottom channel. So in between, we can put skin just to separate the top from the bottom. And then we can feed KK-103 in the top channel. And over time, we monitor how much KK-103 permeates into the bottom channel. And so that is a way to measure the skin permeability of different drugs. And to our surprise, KK-103 does show a really strong skin permeation. For example, this 30 minutes after skin topical application, we got about 60% drug already crossing the pig skin and going into the bottom channel. And by 3 hours, we almost achieved 90%. So this is quite encouraging. And our next step is we want to make sure our animal protocol is approved so we can move into animal study directly. Okay, so I think the final slide, just want to acknowledge many people here, especially KK, who is obviously the inventor of KK-103. He synthesized a small library of those leu-enkephalin analogs, and then did initial tests to identify KK-103. And Roland, also another postdoc in the lab, and he developed analytical methods to help us also screen different analogs and pharmacokinetic studies. And Lucas actually collaborated with Netta doing animal studies to study all the pharmacology effects and also side effects of KK-103. And I also mentioned Brody, who is a new addition to the lab, and he's right now working on this, both projects with Tony and Brian and Puya. Okay, I also want to acknowledge the funding agencies, and thank you very much for your attention.
Kick off with a question about site of action. So when you say CS, central nervous system, of course, that's both the brain and the spinal cord. Have you looked for whether there's a greater effect on the spinal cord than the brain, or how can you resolve that?
Right, we haven't looked at that. Yeah, so it will be great to learn that from you, what kind of assay we can do to really tease out is coming from the brain or the spinal or other things. Yeah, so we are not experts in the field. So we are we know about chemistry, we do drug delivery, but this kind of pharmacological assays definitely we rely on collaborations.
Ask the question is that when you were showing the distribution across different tissue types.
Yeah.
The brain was really very, very low.
Yeah.
I know it can be detected, but it was, how does the amount that you can observe relate to some of the dosages that you use? Can you draw any relationship there? So
yeah, so this is just one dose study. And in this case, we didn't even inject the effective dose because the radioisotope method is very sensitive. So we only need to spike a very little amount of those radioisotope, the KK-103, and then did this experiment, right? So I think it would be indeed interesting to do a dose response curve just to find out whether the accumulation in the brain is also dose dependent.
look at the y-axis on the lower part of A. That's percent. So I'm just looking at the measure, injected dose.
Yep.
Percent dose of, what, how do I interpret that? I mean,
Right.
way lower than the upper part.
Sorry, this?
Yeah.
Oh, yeah, these are the, because we examined a panel of tissues. On top, they are the higher amount of accumulation. In the bottom, they are lower. So brain is lower.
And the point I was trying to make is that the lowest of the low
Mhm.
is the brain.
Mhm.
And yet you still think the sufficient amount is getting into the brain at high enough doses
Mhm.
to map onto all the other beautiful work shown.
I think so, yeah, because we do have this naloxone blocking,
Yeah.
showing that the effect is coming from the central. Yeah, so likely even at this very low amount is still effective. So that I think that also comes to our arthritis project, and also another thought whether KK-103 also has some efficacy coming from peripheral. So we want to also look at that in a different model just to examine that, because I think that will be really interesting if we can induce local effects without going to the brain. And I think that that will be really interesting because this topical application, people are thinking that this is desirable for arthritis because we need local treatment so that we have better efficacy and reduce the systemic side effects.
The other point you were making at the three families of opioid receptors, you were making the point that it's more active on kappa versus, delta versus kappa. Is that the point? Was that the take on that?
Yeah, so that's what we really want to lead this interpretation too. But we understand this. There's some limitations of these inhibitors. They are not pure. They are not specific blockers for DOR, MOR, or KOR. So I think that we can only conclude likely KK-103 is working largely on DOR, but we cannot exclude other possibilities.
So it's the comparison of A being the most suppression.
Yeah.
And that's the delta opioid receptor.
Yeah. Yeah.
Anyway, very nice program.
Yeah.
Are you looking at anything else for that, the end protection?
Yeah, good question. So I think KK later on synthesized another panel, and none of those worked. So we were really lucky getting this KK-103 worked from the first, I think 20 analogs that he synthesized. Yeah, because in the beginning, we really just wanted a very minor modification to the N-terminus, nothing extreme. So he were able to synthesize just a few of those and quickly went into animal trial, right? So I think we got lucky to select that 103 compound.
How much of that's felicity? How much do you think of that is the stability? Right? How much of that
Yeah.
keeps it around long enough?
Yes.
How much that helps
Right. Yeah. So my student did a kind of a correlation analysis, and it's found that O3, I think O2, maybe 3, I don't remember now, it's published. I think 2 factors are very important, both. If we separately analyze and correlate them with the efficacy, they don't have a really good R square. But if we put them together and then compare with the efficacy data, then the R square is much higher. So I think both are important. We don't know which one is more important. Right. But it seems like that kind of modification is enough to improve both.
So why did you choose leu-enkephalin instead of methenkephalin?
Right. So I don't know if this is true, but from the literature, at least in this figure, it suggested that methionine here. So methionine and enkephalin is also activating mu receptor. So because we want to avoid, we want to target delta. So that's why we select leucine.
Because it's more related, well, they're both related to analgesia, but there's much more side effect potential, I guess, in the mu.
Yes. Yeah.
Thank you very much.
Thank you very much.